that you could approach the registration desk upstairs and be provided with a link for directly booking those rooms. So we're going to move now from video games to artificial fish collectives with a talk from Sonja Johnson-Yu from Harvard University. Sonja.
Great. Thanks so much, Evan. Hi, everybody. It's great to be here today, and I'm going to talk about emergent electrosensing in artificial weakly electric fish collectives. And this has been a team effort through and through, and in particular, I want to highlight the contributions of my co-first author, Sakpreet Singh, who was not able to be here due to international travel concerns. And this is ongoing work, so very excited to be able to share it with you all today. Today's going to be all about fish. However, I encourage you to think about it as being a story that's more about social cognition in small collectives of animals. And maybe collective can help me figure out how to use the clicker. Aha. I think we're on the wrong screen. Okay, excellent. So this is a scale at which there are a lot of interesting dynamics, both in terms of competition, cooperation, and communication, whether it's orcas hunting a seal cooperatively, or competitive mating, or collective foraging, which we're going to talk more about today. So let's talk about these animals. Weakly electric fish use electric organ discharges in order to sense their environment. So they produce an electric field using the electric organ that's housed in their tail, and that creates this electric field around them, and they sense perturbations to that field by conducting and non-conducting objects in the environment, which helps them figure out what to eat. They can also use this electrosense for electrocommunication. And this happens over much longer distances than the active sensing happens. So let's see the fish. This is a video that I took at the New England Aquarium. And the aquarist has just dropped a bunch of worms into the tank. It's a bit of a feeding frenzy, kind of like when there's free food and grad students kind of start swarming. One of the things that you might be able to hear, there's this sort of staticky sounding thing, and each one of these clicks corresponds to a pulse that's being emitted by one of the fish. So they're swimming around, eating food, bumping into others, trying not to bump into others. I'll move on to another foraging video. This one is a little bit later on, and the worms have settled down a bit, so now they're digging through the ground. This big fish that's coming in is not an electric fish, but it's going to go ahead and stir up some of the dirt and attract a number of the other electric fish to come start digging around and trying to dig up worms. And you can even see there's a little bit of get out of my way dynamics that's going to happen in the center shortly. There we go. All right, so this is incredibly rich behavior, and we want to understand what's going on here because there's electrocommunication happening, there's electrosensing. And so we want to see, what's happening in the social foraging setting. And the way that you normally go about studying that is by taking a number of fish, putting them in an experimental tank, and then seeing what they do. But there are a number of challenges of really understanding what's going on here. For one, if you want to get a naturalistic behavior, you're going to need to have a naturalistic environment. And you want that arena to both be naturalistic, but also you want it to be controllable so that you can run manipulations on it. You might want to do certain like sensory or motor interventions, which can be costly. And finally, doing multi-animal neural recordings is often infeasible or very challenging. So our proposition to solving this and understanding these fish a little bit more deeply is doing virtual social neuroethology. So we're creating artificial agents in simulation because they're easy to manipulate and we can have full observability over their behavior and their neural computations. There's precedent for these agent-based models inside of neuroscience. We've got these two-armed bandits that reveal many of the components of decision-making. And on the more embodied side of the spectrum, you've got these actual body models of virtual rodents and mechanical flies that are running around in Mujoko environments. And then in the middle, you have this sort of this medium level of abstraction where you are using point-based agents and things are happening in 2D, but there's some detailed environmental modeling and detailed sensory modeling. And right now, what you're seeing in the middle is the PhD work of my co-first author, Sat, who simulated flies doing turbulent plume tracking and made some accurate predictions about how flies actually implement this. What we do is we're working on the same sort of intermediate level of abstraction, and we build on the sensor capabilities of the agents and then expand to the multi-agent set. So we've got this marvelous toolkit at our disposal, and I'm going to go through it one component at a time, starting with the environment. Because this is being done in simulation, we have full control over where the food is, what the distribution is like, when the food runs out, how quickly it replenishes. And then we also implement electric field generation simulation. So whenever a fish chooses to pulse, we simulate the dipole that's produced by that fish. We also simulate the induced dipoles. This is how real fish are able to tell that that thing's a worm because of the induced dipoles. And then finally, the reflections on the walls of the tank. And on the right-hand side, on the bottom, that is our arena and a little snapshot of the electric landscape. And then on the top, that's in comparison to actual experimental results on electric fish. Next, the agent is running off of an RNN backbone and feeds to actor and critics. The RNN is because real brains have recurrence, and we want to mimic that as well. And then the agent also comes with an electrosensory model that we build. So real fish have 3 different types of electroreceptors. They've got their M sensors that are used for active electrolocation. They've got their A sensors that are used for passive electrolocation, so they don't have to pulse. They can just sense these the fields of the worms nearby. And then they've got these long-range passive electrocommunication K sensors, which facilitate that social communication. And we implement the same types of sensors on our agents. And specifically, we mimic the spatial distribution, the relative ratio between the different sensor types, and also we've carefully tuned them so that they have the actual same sensor thresholds that you see in the real fish. So these get concatenated together into sensory observation vector and also add in like some internal variable information, for example, like the size, which governs dominance in these fish. They can move and turn continuously and then choose to discretely either pulse and bite at each time step. And then we choose a very simple reward function, which is eat food and don't get bitten. So that all is optimized using multi-agent PPO. And when we put it all together, we get this emergent collective foraging behavior. So the gray lines are the fish pulsing. They're able to traverse the arena. On the right-hand side, you can see those are trajectories at the end of a foraging episode where the X's are eating. They avoid each other so they don't get bitten. And they pulse. And that's something that we can plot as well at each time step. We can also plot the distribution of the interpulse intervals and see that it shares like power law-like characteristics that you see in real elephant fish. So in summary, we built this framework. We've got variability in training. We've got agents that share the same capabilities that real fish have, and a reward that's guided by fitness. And they learn to forage for food, explore the arena widely, and avoid aggressive interactions. So how can we further validate that these agents are really behaving like real fish? To answer this question, we take inspiration from a classic experiment by Hopkins, where there's one fish that's fixed into place and another fish that is doing this sort of homing behavior. It's trying to approach the other fish. And we want to see how our agents solve this problem. And what we see on the right-hand side is that they take these sort of circular approaches to get to the fixed agent. We compare this with a baseline. We implemented a non-electric sensing model that is doing more of a ray casting, like LiDAR-like thing. And this one takes these straight paths. But our electric field model propelled agents, they're taking these circular paths. So let's dig into like what's going on there. We're going to look at the error angle, which is the difference between the electric field direction and the agent's orientation. And if we plot that error angle over time, this is over the course of the episode, we see that at the beginning, the agent might be oriented any which way with respect to the electric field. But over time, they get to orient themselves either parallel or anti-parallel to the electric field lines. How do real fish do it? Turns out they solve it the same way. You can see the same modes at the parallel to the electric field and the anti-parallel. And because we're working in simulation, we can look into the neural representations of the hidden state of our RNN and see what's being represented. So from that hidden state, we're just going to predict some task relevant variables to see which ones are most clearly encoded. And what we see is that indeed it's this error angle to the electric field that is encoded fairly strongly in comparison to like the distance to the nearest agent or the angle to the agent, which is not representative whatsoever. So in summary, from this homing experiment, we can see that by constraining the capabilities of our agents to this known biology, we can produce realistic behavior, even in the absence of new experimental data. We didn't have to do any fitting here. Now I'm going to talk about two virtual experiments that we're doing. So we're going into a social foraging assay. These fish have been trained in a general foraging case, and by fish I mean agents. So we've got the fish A, which starts inside of the patch. So it should be happily eating. And then we initialize B randomly somewhere where it's within communication distance to fish A. And then what we can do now is we can vary their relative dominance level. So this is one of the internal variables that we introduce. And what that means in practice is that changes how much it hurts to be bitten. Like if the size difference is small, it looks like this. Size difference is big. Ow. What I'm plotting right here is a bunch of trajectories in these different dominance conditions. And one of the things you see here is that B, that's plotted in sort of orange red, it swoops in, it takes over the patch because in this case, B is dominant and ousts A. And then in this case where they have the same dominance level, they're just kind of fighting over the patch. And then in this case, A dominates the patch and so on and so forth. So it's not trivial for B to be able to get to the patch. So we can also plot the number of episodes where B gets to the patch. And we can see that that correlates with B's relative dominance level. So let's think about this case where we get rid of A entirely and we just have B. B's free to eat, B's free to find the patch and eat. Do we think it's going to be higher or lower? Raise your hand if you think it's going to be higher. Raise your hand if you think it's going to be lower. People don't have many thoughts, but some people think it's going to be lower. And indeed, it is lower. And that's because B is leveraging social information in order to find the patch. B is leveraging information from A in order to figure out where the patch is. And then on the right-hand side here, we can see that it's not just relative dominance that is governing this behavior, but also B's random initialization, how far it is from the center. That's controlling it. So when B is dominant, it can find the patch even if it's really far away. If it's subordinate, it only has a shot if it's close by. Now, one last experiment. What happens when we remove their capabilities to do electrocommunication? So we're going back to this 4-fish setting, and we're knocking out the K sensors, which are used for electrocommunication and sensing those long distance EODs. What happens is that we see that the foraging rate drops. They eat less food. And in fact, this result has been found in zebrafish. Similarly, that social foraging is better than foraging alone. Other things that we find that the biting incidents rise because they're using these social sensors to avoid each other. And their movement bouts get longer because they no longer have social information, so they need to explore more. So these are some of the predictions that we're making, and we're in conversations with Nate Sawtell's lab at Columbia to figure out what real experiments they're actually planning on doing. I have some fun analyses that attention-based interpretability analyses that you can also see in my poster. So the summary is that we've put together this framework for modeling the collective behavior, validated the model, and I've shown you a couple of early vignettes of virtual experiments and analysis. So we're doing more virtual experiments. We're probing into their neural computations, and we're thinking of this as being an experimental co-pilot, which is used to generate hypotheses for future experiments and whittling down the hypothesis space. Last thing is that Sot is on the job market, and he really hoped to be here and connect with you all. But because he wasn't able to be here due to international travel, I strongly encourage you all to check him out on social media and come chat with me at Poster 3. Thank you.